it's amazing what you can do. So sometimes your imagination is the only limit.
Welcome to Called to Science, where we dive into the untold stories of the scientists pushing the boundaries of the life sciences. I'm your host, science journalist Brad Van Perdon, and today we talk about the importance of balance, the creativity of bacteria, and our guest's favorite microbe. Hello, Joleen Masschelein.
Yes.
I got it right.
Perfect.
Okay. This podcast is titled Called to Science, and I think only someone who is truly called to science would get the idea to isolate bacteria from their student restaurant and discover a new antibiotic. And this is exactly what you did. And amazingly, this isn't the only antibiotic discovery you've made in your still young career, as you also discovered an antibiotic agent against one of the most dangerous multi-resistant bacteria in hospitals and co-developed the basis for a new antibiotic for drug-resistant tuberculosis. Now, as a junior PI at VIB, you continue your work to harness and explore the biosynthetic creativity of microbes, all while mentoring a new generation of researchers. Welcome, Joleen, to Call to Science.
Thank you very much. Happy to be here.
Well, I'm happy that you're on the job because bacteria beware. You're doing a lot to protect us against infections.
Yeah, it's amazing how many cool molecules bacteria make. And back in the days when I started as a master's student, I was surprised how some of our most used antibiotics in the clinic are actually coming from bacteria.
Mm-hmm. Yeah, in my undergrad, I remember learning this and was sort of blown away and kind of blown away at the precarious situation we find ourselves in. And we can get to that in a little bit because I do have several questions about bacteria. But I first want to start with your introduction to science and the first time maybe that you thought, hey, this could be a career.
I think it always inspired me to see amazing scientists like Catherine Verfaye, who was making the news with the discoveries on stem cells. And it was always very, very inspiring to see. And I think that sparked the first interest for me in science and a scientific career. But back in the day, I never thought, I would be capable of a career like this. It's something you kind of roll into as you start to study more, as you go to university and you learn more and more, and you do practical labs and you discover new things, and that's how you slowly roll into it. And the more you discover about, in my case, the natural world and bacteria, the more I was interested in investigating further, and that's how I got started in it.
I'm curious, specifically microbiology, bacteria, what drew you to that? Because for a lot of people, it's gross. Bacteria equals gross. And for me, my academic pursuits was kind of similar. I was really into infectious disease and parasites. But for me, there was a moment when I was 11 years old that really got me started on this track, and it was a 90s movie called Outbreak.
Oh, really?
I don't know if you know the film, but looking back, a little cheesy, but the villain of the movie is an Ebola virus. But as an 11-year-old, this terrified me, but also fascinated me and put me on this trajectory where I've been interested in this stuff ever since. Was there some kind of moment where you were just like, bacteria, everyone else thinks this is gross, but wow, I'm just fascinated by it?
I think during my master thesis project, where we had discovered a new antibiotic made by bacteria, I think that for me made me realize that bacteria do so many useful stuff for us. And actually there's so many beneficial bacteria that live with us all the time, that do important functions for us. But I think that thesis project, the fact that we could get new antibiotics from bacteria, I think that really triggered for me the interest in this topic.
Mm-hmm. So again, kind of sticking with this similar thing, and maybe you don't feel the same way, obviously, because you work in this, but do some of your family and friends, are they sometimes curious as to what are you doing looking in the microscope, looking at these weird little bacteria all the time?
Yes. Yeah. They often ask me what I'm doing, and then I try to explain it in the most clear way. And I do. I mean, they are typically always amazed about these things. So I'm always happy if I can convey my enthusiasm to my family and friends as well.
Yeah, I think a lot of people are starting to realize the beneficial side of bacteria, right? With the interest in the microbiome and everything like this. Because, yeah, I think maybe 10, 15 years ago, our only thought about bacteria would be, I have an infection, I need to get rid of this, bacteria bad. But we're starting to come around to the beneficial side. And there's this whole unexplored world. How exciting is it for you to kind of be maybe an ambassador for the good side of bacteria?
Yeah, it's really interesting to see what bacteria can do for us. Not only their natural capabilities, but also we are getting so much better at engineering them to make them even more beneficial to us, or to give them new functions, new tools, so that they can do useful things, or make useful molecules for us, help treat human diseases, communicate to us, signaling that there's something wrong. I mean, we can exploit bacteria in so many different ways because they are actually brilliant chemists. They are among the most brilliant chemists on this planet. They're much better at chemistry than we are. If we go to the chemistry lab and we try to piece together a molecule from scratch, we are so inefficient. It takes us months, depending on how complex the molecule is. But bacteria do it effortlessly. And so we can really engineer them. We can use their skills so that they make useful molecules for us, not only for health, but also industrially useful compounds like biofuels or other sustainable, other molecules that are nowadays produced in a very laborious, expensive, inefficient way. They can do it much more sustainably for us.
Some of your work, you're working with these bacteria that live very closely with a host, or they rely on a host. And there's something about these relationships, whether it's symbiotic or commensal, parasitic, sometimes all at once, depending on the environment. Something that to me seems very fundamental about life, these relationships. Do you think about that? And is there something to be learned about why life has decided to organize itself in these relationships, these interconnected relationships?
Yeah, it's fascinating to me how they live together, how they also communicate with each other in a language that we're only just starting to interpret. And in the end, each organism is also invested in its own survival. And like humans, it will undertake interactions, helping a friend, for example, if that's beneficial to them, or fighting off a common enemy. Depending on how it can promote its own survival, it will find allies, it will organize itself into a community, it will try to establish itself so that it can have enough access to nutrients. And it's a bit like humans do. In order to survive and to feel good, they will also make friendships or fight a common enemy, and so it's much similar to how we behave, I think.
You've mentioned this a couple of times now, but the ability of bacteria to produce useful molecules for us, and this is a lot of what your work is focused on, is finding some of these useful molecules. So let's start with the idea to sequence bacteria from the student restaurant. Was that just a shot in the dark? Did you get lucky with that one? Or did you have some idea that maybe there was something here?
So I only came into that project when the bacteria was already isolated.
Okay.
Another colleague of mine at VI-KU Leuven, who is a professor in food microbiology, he sampled student restaurants, vegetable cutting surfaces, to see what kind of microorganisms live there. And among the bacteria he found was one that made this broad-spectrum antibiotic. And that's when I came in for my PhD project to figure out, what is this antibiotic? How does it work? How do the bacteria make it? And it turns out it was made in a very unusual way.
These biosynthetic pathways and, developing them and engineering bacteria to then produce the thing that we want, the thing that we're interested in, antibiotic or things for the chemical industry. I don't think a lot of people outside of research, really care about what happens to the bacteria in a lab. But do you ever think about it from a bioethics standpoint, that you're engineering life? It's kind of quite profound, I think.
Yes. Of course, it's very important to do this in a safe and contained way. And this is something the university also makes you aware of and makes you comply to a high degree of safety, biosafety then, just to make sure that none of these engineered microorganisms escape into the environment or things like that. So we are very careful about this. So they don't typically leave the lab environment.
Mm-hmm. But it must feel, I'm thinking there must be some kind of a thrill at being able to sort of engineer a living thing at that level, with that level of detail. It's really quite cutting edge stuff.
Yes, and it's amazing what you can do. So sometimes your imagination is the only limit. Yeah.
So I've written a few stories and talked with synthetic biologists and chemists who have done some of this work, making novel pathways so that bacteria can create things for the chemical industry in a more sustainable way. But they've told me that just because it's possible and that they can do it in the bacteria, there's an extra hurdle of actually getting industries and broader applications to take up this technology, disrupting the current production methods and stuff. And they say that they need a real political and even financial backing to help get some of these technologies off the ground. Do you have a thought on this, or how do you see bringing these things from the lab to actual real world applications?
I think that's where VIB comes in and provides a lot of useful and valuable expertise in bridging kind of the gap between academia and industry. For example, I have now collaborations with, for example, Johnson & Johnson, using microbial tools for making peptide drugs in a sustainable way. I also have other collaborations with companies that are already interested in using microorganisms to make clinical therapeutics. They are already using them, but they want us to make the bacteria more efficient in making those compounds, so higher efficiency. So I think industry is already there, is already interested and is already using microorganisms more and more to do these processes in a more sustainable way. But interest is increasing as we speak because of the current economic landscape as well. And because it's important to try and be more efficient, sustainable, and if at the same time you can cut costs, then yeah, it's a no-brainer to use microorganisms.
And now it's time to dig a little deeper in a segment we call Dare to Ask. So I'm going to ask you some questions that people would like to ask but have never really dared to ask. And these are questions that come from the VIB community. Are you ready?
Yes.
Okay. How should I approach my group leader to ask for advice?
Yeah, don't be afraid. Come to me, talk to me, and I'm happy to help. Yeah.
What is the biggest myth about being a scientist at VIB that you would like to debunk?
Maybe that everything is extremely competitive and you have to work so hard all the time and have no life at all. I think that's certainly not true. Everyone is very supportive and is so willing to help each other. And there is a lot of attention to healthy work-life balance.
Great. How would you, or have you, handled a disagreement with colleagues about your research?
Oh, I don't think I've really had a big disagreement yet about research. And if I would have, I would listen to their arguments. I would hope the other person would listen to my arguments, and we figure it out together. It depends what kind of disagreement, I guess. Is it really a scientific disagreement or?
I don't know. My sheet doesn't say, so I guess we're free to. We're free to. But I think. I mean, I would think of a scientific disagreement because scientists tend to get very passionate about their answer or their approach or something like this. So how do you navigate maybe those sorts of discussions?
Oh, scientific disagreements are fun because they make you think about, the actual problem and try to find solutions. Think outside of the box, maybe. Because that's usually when something is not clear or evident. That's usually when it's becoming very interesting. And then you listen to different viewpoints and you learn new things.
If after listening to this Dare to Ask, you find yourself with a question you've never dared to ask, let us know in the comments. And who knows, maybe your question will end up on the next episode. Maybe taking a step back and looking at sort of your research and your practice of research, you have a lot of different things, it seems, going on in your lab. How do you balance switching from, the niche, looking at one project, one bacteria, and then also the big picture, the larger picture of what you're trying to accomplish with your work?
So all of the projects in the lab are kind of interconnected in a way, centered around these fascinating biosynthetic capabilities of microorganisms. But it's really interesting not to have to focus on one project, but to explore many different applications. That's what makes it exciting to me. And to always think strategically for new projects and new areas of application.
Yeah, because I think I'm thinking too of, I've often heard a criticism of science right now is that people get too siloed, they get too focused on one thing, and then you miss those opportunities for collaboration with other labs or even other disciplines. So how do you maintain, focus on what you're trying to do, but also be open to the other things that are going on? I mean, it's just a lot to keep up with.
Yes. And I'm also someone who naturally has a lot of interests in many different things. But actually, what I really like about our research is that it's very multidisciplinary. We combine chemistry with microbiology, synthetic biology, enzymology, structural biology, bioinformatics. So we naturally have a lot of collaborations with our lab. Many of my students have co-supervisors in other labs, in other departments, in other groups, in other universities, in other countries even. And I really like this big network because it helps you really lift also research to the next level if you can collaborate with many people. And naturally, by collaborating so much, you learn about their research. And it's inspiring because sometimes you can get new ideas that you wouldn't have thought of just by yourself. But because you listen to your colleagues and your collaborators and you learn about what they are doing, you think, is there something that we could do at the interface and that could lead to unexpected ideas or projects that might not be just evident like that.
So talking about balancing the big picture, balancing the niche, how do you balance a busy lab and a busy research career with the rest of your life?
So it's important, I think the key word is balance, right? And it's important to keep track of that balance because if you don't, then you will not be able to keep doing this. So for me, it's important to family and friends always come first. And I always make sure that I plan enough time for hobbies as well. And then around that, I plan the work. But there's certain things that I plan first and then the work around it, in the free time, I mean.
I think that might be a bit surprising and maybe refreshing for especially younger career scientists and graduate students to hear, because I know when I was in grad school, the pressure to be in the lab all the time, to get your data, to be working, it was almost felt like a competition at times. But to hear someone who's made a successful career out of it say, no, you need to have that separation, that boundaries is quite nice to hear.
Yeah, it's important to realize that you only have one life and life goes by very fast. Days fly by, right? It's important for you to figure out what are your priorities in life. And also what gives you energy, and to make sure you have enough things in your life that give you energy and not just drain energy. I think it's important to keep that balance. And that is very different from person to person. I mean, some people just get all their energy out of science, and that's also fine, right? But for other people, it's important to have other things as well, just to make sure that it stays a healthy career.
Here's one that's maybe a bit left field, but you're very interested in microbes. They're hardy organisms, let's say. They can survive in all sorts of different environments. Do you follow the news of microbial life in space, the possibility of microbial life in space?
Yes.
How exciting is that for you?
It is very exciting. I mean, yeah, if I would immediately look what kind of molecules they would make and things like this. But of course, what would that look like, right? It might be different from how we define a microorganism on Earth. It will definitely be exposed to vastly different environmental conditions than here on Earth. But that's super fascinating, right? Like, how will those microorganisms have evolved to live in those environments wherever they are, you know? I'm sure, it will be very fascinating to see how they look like and whether they have the same basic building blocks as microorganisms on Earth, or whether they have evolved completely different strategies to live and replicate.
So the way that you were speaking about it there sounds like you believe that there is microbes out there. I'll put on the table, I think there's probably something out there.
Yeah, let's say that I wouldn't be surprised. Mm-hmm. Yeah.
Would you, if you had all the funding, all the money in the world, would you want to go out and try and find microbes in space? Or are you happy to be here on Earth?
I think I would use the money to send someone and then bring it back here to me.
Okay. Then in the vein of sending someone, you're now running a lab, you have students of your own, you were a grad student, you traveled around to a few different labs. What's something that you learned along your academic journey that you try to instill for your students now?
I think one of the things that really worked for me and didn't come natural was being proactive and being assertive. Because opportunities typically don't just fall on your lap, but it's surprising how much you can achieve if you go after something you really want and if you put effort in it and chase it. A whole world of opportunities opens up if you put some effort into it. And I think, I would say to anyone, never be afraid or scared to go after something you want. Be proactive, be assertive, and then, a lot of cool things can happen.
Okay. You work with a lot of different bacteria. Do you have a favorite? Or let's say favorite microorganism, if we go broaden it a little bit.
Maybe Pseudomonas bacteria.
Okay. Describe it for us, for those of us who've never heard of this before.
They are really interesting in the sense that they produce a lot of fascinating molecules. We recently found new anti-cancer molecules from Pseudomonas bacteria isolated from a fish disease outbreak. But in the past, I've also worked on an antibiotic from a Pseudomonas strain that was active against the very notorious human pathogen Staphylococcus aureus. Mm-hmm. And it's interesting how versatile they are. You can find them in a lot of places, and they typically make interesting and useful compounds for us. So, and also importantly, it's fairly easy to genetically manipulate them. So they listen to us.
They're easy to get along with.
They behave in the lab.
Is there one then that doesn't behave in the lab, but that you're like, wow, if we could just get something here, you know that there could be some real opportunity there?
There's some bacteria, and when we look at their genomes, it's like a treasure trove of new molecules that are likely having interesting biological activities, antibiotics, anti-cancer agents. But it's so difficult to, on the one hand, grow them, and on the other hand, engineer them to unlock these molecules, to identify them.
As we kind of wind down the conversation here, I'm curious, you spent some time in the UK, I believe, during your postdocs, and then you came back to Belgium and are now at VIB. What was it about the opportunity at VIB that really spoke to you?
You know, when I was in the UK, I was in a lab that was very multidisciplinary. And in the lab, there were chemists, there were biologists, there were bioinformaticians, there were structural biologists. And it also had all the infrastructure in all these disciplines. It was all there. So that was amazing to me because I could just focus on the research and learn so many new things from my colleagues who were from other disciplines. And everyone helped each other and everyone learned from each other. So when I came to Belgium, I wanted the same. And VIB is an institute that allows me to do that, to combine all these disciplines. And that's really what I also need for this type of research. You can't do this if you're only focusing on biology. You need these other disciplines. And that's something that VIB really allows you to do. And it also provides us with access to cutting-edge technologies so that we can really focus on the research and don't have to worry about the tools or the instruments needed to do that.
You started at VIB, you're one of the younger PIs now here. When you first started, did you feel any sort of pressure from the older labs, the reputation that they might bring with them? They're more established.
So it's definitely very impressive to be among those amazing scientists, and with all their achievements and their impactful work. But to me, it was very inspiring. It really helped me. It gave me energy to start my own lab, give it my best, and also try to make a difference that way.
So something to strive for rather than be intimidated by.
Yes, absolutely. And everyone is very supportive and tries to help you. VIB as well, they try to help you in any way they can to start up your lab and get started. Yeah.
And what about some of the other new PIs or other junior leaders? Have you guys formed kind of a community that maybe helps support each other in this as you're all starting out?
Yes, indeed. For example, when we started VIB, we all have to follow this leadership course. And that's where we meet a lot of our colleagues who also recently joined VIB. And we keep in touch. And if we have questions for each other or advice for each other. At the same time, I'm also, for example, chair of the group leader committee at VIB, together with another colleague of mine who started around the same time as me. And that's also really interesting to see how VIB works and to get to know our colleagues at other universities in Ghent, in Antwerp, in Brussels.
Great. So before we finish up, we'd like to ask a final question, and it's the same question we ask every guest. And that is, if you were to encounter an omniscient being and could ask one question, what question would that be?
I would maybe ask how it all started.
Life?
Yes.
The universe?
The universe.
The big question.
Yes.
It's a good one. All right. Joleen, thank you so much for the conversation. It was a pleasure.
You're welcome. Thank you.
If you want to hear more inspiring stories from our scientists at VIB, be sure to subscribe to this podcast. And if you enjoyed the last episode, leave us a like or positive review. It really helps. Thanks for listening to Called to Science.